What's going on, everyone? If you don't already know me, my name is Will Kitzmiller, and welcome to my DNA structure and replication project. In this video, I'll be showing you the 3D project I created in Minecraft and talking about some key scientists in biology's history. Now, let's jump straight into it. Alright, so first I'm just going to give you a quick look at my entire structure from different angles, and I'm going to dive into explaining it. Now, let's look at the three-part structure of a nucleotide. I'll start with this pinkish block, which represents the phosphate, and the purple block, which represents the sugar. This yellowish-orange honey block represents the covalent bond between the sugar and phosphate, so this all shows the sugar-phosphate backbone. Now, let's look at these colorful blocks in between the backbone. I used red, blue, yellow, and green to represent the base pairing of nitrogenous bases. The yellow block is adenine, and the green block is thymine. These always have to pair with each other. Then, the red block represents adenine, while the blue block represents guanine. And these two always pair with each other. The black glass in between these represents the hydrogen bonds they create. So pretty much, the sides are the sugar-phosphate backbone, the middle is the nitrogenous bases, and this structure is all held together by bonds. Now, as we move up my model, you can see the helicase, represented by these black blocks that is unzipping the DNA strand and unpacking the genetic material. The replication fork occurs when the helicase happens, so pretty much the helicase splits the DNA, and the DNA begins to take the shape of a fork. Now, at both ends of the fork, there is a DNA polymerase, which I represented with white blocks. I only made one DNA polymerase, but there's always like one on both sides of the split. This ring creates new strands of DNA that are exactly identical to the original one. So the purpose of these DNA polymerases is to create two new identical DNAs. And that's pretty much my 3D model. Alright, so now for the boring part, but very important, I'm going to head into the elaboration on some concepts. Let's start off by talking about the works of some famous scientists. I'll start with Alfred Hershey and Martha Chase, the dynamic duo in the field of molecular biology. In 1952, Hershey and Chase conducted an experiment that provided evidence for DNA as the genetic material. In their experiment, they used a virus that infects bacteria called bacteriophage by labeling the DNA as the phosphorus and the protein coat with a radioactive sulfur. With this, they were able to track which component was transmitted to the next generation of bacteria. The results showed that the DNA was passed on, not the protein. This was a groundbreaking discovery in the field of genetics, as it proved DNA carries the genetic info. Alright, so moving on from Hershey and Chase, James Watson and Francis Crick were the people who discovered the structure of DNA. They created a model of a DNA strand that was made of two strands that twist around each other like a spiral staircase. This allowed scientists to understand the structure of DNA was a double helix, which changed our understanding on genetics forever. Their discovery was game-changing as it laid a foundation for modern molecular biology and how we understand how genetic info was stored and transmitted. Now let's talk about Rosalind Franklin, the scientist who took the very famous photo in 1952, Photo 51. She conducted work in X-ray crystallography, and these X-ray images of DNA provided very important insights of its structure. Photo 51 showed the helical nature of DNA. Watson and Crick used her data to eventually propose the double helix structure of DNA. Even with her massive contributions to molecular biology, she did not receive the credit she deserved at the time, which is a very controversial topic to this day. And last but not least, Erwin Chargaff was a biochemist who conducted experiments to analyze the base composition of DNA from different species in the 1940s and 1950s. He found out that among the adenine is roughly equal to the amount of thymine, and the amount of cytosine is roughly equal to the amount of guanine in DNA, which became known as Chargaff's rules. This discovery provided evidence for base pairing in DNA, which was later put into Watson and Crick's double helix model. So finally, I'm going to elaborate on the purpose of DNA replication, a very important process that occurs in the nucleus of cells. It happens when a cell is preparing to divide and create new cells. The purpose of it is to make sure that each new cell receives an identical copy of the DNA. This process occurs in the S phase of the cell cycle and involves unwinding of the double helix of the DNA, the strands separating, and the synthesis of the new strands. This process allows for the growth, development, and reproduction of all living organisms. And with that, that's the end of my project. I hope you enjoyed, and thank you for listening.